Welcome to Biogas Basics. My name is Jürgen Kube. This is the fifth and final presentation of this mini-series about microbial growth. Here, I will show how the different phases of the AD process all work together and why even severe inhibitions and limitations are not necessarily a problem for an AD reactor. I will then conclude the mini-series with a brief recapitulation of the former presentations. Now let's put it all together. In the first part of this mini-series, I talked about general correlations between growth rate and dilution rate, or residence time and doubling time. In the second part, we discussed the various hydrolysis kinetics. The hydrolysis is often the rate-limiting step when fibrous feedstocks are digested. In the third presentation, we talked about the inhibition and limitation kinetics of methanogens. The fourth presentation dealt with the thermodynamic constraints of acetogens. If we add it all together, we get a simple model of the biogas process. Complex feedstocks are broken down by acetogenic fermenters to hydrogen, CO2, acetic acid, and higher volatile fatty acids. These fermentative bacteria make the hydrolytic enzymes that are required to convert biopolymers into oligomers, di and monomers. The hydrolytic step is often the rate-limiting step, especially when the feedstock comprises large and insoluble biopolymers. The metabolic end products of acetogens are mostly hydrogen, CO2, volatile fatty acids, and ammonia. Hydrogen, CO2, and acetic acid can be directly degraded by methanogens. Higher volatile fatty acids must be converted by syntropic acetogens to acetic acid, hydrogen, and CO2, which then can be consumed by methanogens. Higher volatile fatty acids, especially propionic acid, inhibit the methanogens in a non-competitive form. Ammonia inhibits methanogens as well. Hydrogen, and to a lesser extent acetic acid, inhibit the syntrophic acetogens by making acetogenic reactions endergonic. In a normal steady state, the concentration of the intermediate products such as acetic acid, propionic acid, and hydrogen are very low as they are consumed by the microorganisms further down the process chain. But when the system is transient, for example after a hydraulic or feed shock, the different stages can react with different speeds. The fermentative bacteria usually show the highest growth rate, or a Mumax, or the lowest minimal doubling time, which can be down to an hour. Methanogens grow a bit slower, showing doubling times in free growth of several hours. Syntrophic acetogens are the slowest to react. Consequently, their adducts, higher volatile fatty acids, are the first to accumulate in these conditions. When hydrogen accumulates temporarily, homo-acetogens can convert it to acetic acid, but propionic acid cannot be oxidized at high hydrogen partial pressures. I will quickly discuss other limitations, most notably micronutrients. Methanogens and acetogens have highly conserved enzymatic pathways that rely on metals as cofactors or use metal enzymes right out. Cofactors are smaller molecules that carry functional groups from enzyme to enzyme, for example the methyl group in the methanogenesis. Metalloenzymes are enzymes that have a metal ion integrated into the structure of the enzyme, usually forming a metal complex. Cobalt and nickel appear in the cofactors and are mandatory for methanogenesis. Iron is used in several enzymes catalyzing oxidation and reduction reactions, and is also integral in ferredoxin and hemes, which are molecules used in electron transfer. Usually, trace elements are bought as a ready mixed solution and added to meet the minimal requirements of the digester. Individual dosing to certain target levels is less common. Trace elements can precipitate with sulfide ions in the digester, so they are typically added to the digester in a complexed form, as EDTA, NTA, or citrate solutions. Methanogens and acetogens rely on proton motive force and sodium motive force to generate energy. Sodium and potassium ions are required for this. Some feedstocks can show deficiencies for sodium and potassium. A non-optimal growth temperature can also restrict the growth rate of microorganisms. All microorganisms have an optimal growth temperature, but if the temperature is a few degrees below or above the optimal growth temperature, the microorganisms can still grow, but show a higher doubling time in free growth. Usually, microorganisms are less affected by a negative deviation from the optimal growth temperature than by a positive deviation. Classical temperature optima are clustered around 5, 20, 30, 37, 55, and 65 degrees Celsius, which stand for certain biological niches. Above that, temperature optima are arbitrary and not clustered. While hydrogenotrophic methane formation is possible at hyperthermophilic conditions, biogas reactors struggle to work above 60 degrees Celsius, as acetate can no longer be consumed by acetoclastic methanogens. A deviation from the optimal pH value also slows down the growth rate of microorganisms, though microorganisms show a wide range of pH optima. especially microorganisms that can generate energy through substrate-level phosphorylation, or SLP, which are the acetogenic fermenters and some acetogens. Microorganisms that rely on electron transfer phosphorylation, or ETP, generate energy by maintaining proton or sodium gradients over the cell wall and usually struggle at extreme pH values. Some acetogens and all methanogens fall into this group. Salinity is another environmental factor that can affect the growth rate. Individual microorganisms have adapted to specific environments. Typical salinity optima are around 0.9% and 3.5% sodium chloride, which is the optimal salinity for marine microorganisms. Generally, all inhibitions and limitations can be included in the growth formula. Every inhibitor will add an inhibition term to the formula, here written as I. Ideally, the term is 1 and doesn't impact the growth, but even if a single species, for example nickel, is limiting, it will slow down the growth rate. When we observe a growth rate in a digester, we unconsciously include all the limiting and inhibiting terms into the μmax term if they are kept constant or are not measured. When a paper reports a μmax, it is the maximal observed growth rate under the conditions used in the research. Further media optimization might lead to higher growth rates and lower minimum doubling times for the same species. In a realistic biogas reactor, those limitations and inhibitions can slow down the growth significantly, so that we can assume minimum doubling times of several days even though the microorganisms can double in a few hours under optimal conditions. Before I wrap up, I want to examine what these inhibitions actually do to the microorganisms, and why slight inhibitions are not that bad for the stability of a biogas reactor. Let's go back to the monokinetic and plot the biomass and substrate concentration over the residence time. We see the washout point, the turbidostat, and the chemostat region. Biogas reactors are operated in the chemostat region. In this example, the minimal doubling time of the microorganisms is set to 4 days. The theoretical washout point is at 6.4 days residence time, but this is just a theoretical value. The CSTR will become unstable in the turbidostat region long before it will reach the washout point. We operate the reactor at 30 days residence time. The substrate conversion is 97.6%. Now we induce less favorable growth conditions, for example by increasing the temperature of the reactor by 5 degrees Celsius or increasing the ammonia concentration. The growth slows down by 50%, and the microorganisms now double every 8 days in free growth. We see a slight increase of the substrate concentration. The microorganisms must offset the reduced growth rate with a higher substrate concentration, but the substrate accumulation is marginal. Translated to the AD process, this means that volatile fatty acids start to accumulate slightly under unfavorable growth conditions. As a result, the substrate conversion drops slightly to 93.7%. At this point, we are still in the chemostat region, and even though the inhibition halved the maximal growth rate, the substrate conversion dropped only by 4%. A 4% drop in conversion efficiency in the primary digester can hardly be measured in a commercial biogas plant. Only when we further restrict the growth of the microorganisms and halve the growth rate again to a doubling time of 16 days, for example by omitting a necessary trace element, the impact on the growth rate is now so severe that the microorganisms must tolerate high substrate concentrations to keep up with the required growth rate. We now enter the turbidostat region and the growth would not be stable. The substrate conversion drops to 66%, and a significant amount of substrate will not be converted. Remember, the doubling time of microorganisms in a steady state, CSTR, is the natural logarithm of 2 times the hydraulic residence time, or 21 days in this example. The minimal doubling time is now 16 days when all the inhibitions are considered, and the theoretical washout point is at 25.4 days. This figure shows the binary nature of a biogas reactor. The first 50% inhibition of the growth rate had only marginal impact on the conversion efficiency of the primary digester. After the second 50% inhibition, the plant is not less effective, but simply acidifies. Process inhibitions do not matter until they do, at which point the digester stops working at all. There is no gradual transition from the uninhibited to the inhibited state, but an abrupt step change. A well-designed plant never gets near the washout point and does not require intensive process control. But if the plant operates with varying inhibitions and limitations, meaning that the washout point moves around frequently and comes near the residence time, process monitoring and observation is crucial because if acidification starts, it will happen suddenly. In this case, model predictive process control, for example with the ADM1 or AI-based time series forecast models with exogenous inputs, becomes important. Personally, I favor engineering the problem out during the design phase over applying intensive process control methods to keep a plant alive. But these methods do have value if an existing plant operates far beyond its design capacity. Just remember, for the design phase, stay away from the turbidostat region. Let's summarize what we have covered in this mini-series. We started with the basics of exponential growth in batch cultures. In free growth, microorganisms show a characteristic doubling time. Mathematically, the growth is described with the growth rate μ, which is d ln x over dt. Mu is linked to the doubling time via the natural logarithm of 2. The growth rate is ln of 2 over the doubling time and vice versa. In free growth, the microorganisms can grow as fast as possible. In a continuous well-mixed digester or continuous stirred tank reactor or CSTR, the growth rate is linked to the residence time when the reactor operates at steady state. The residence time is the inverse of the dilution rate d, and, in a steady state, CSTR, the growth rate μ is the dilution rate d. This is equivalent to that in a steady state digester all microorganisms double with ln of 2 times the hydraulic residence time. The growth rate depends on the substrate concentration. The monod kinetic is a simple form of describing this dependence. At low substrate concentration, the availability of the substrate is rate limiting, and the monod kinetic is a first-order reaction. At higher substrate concentration, the ability to transport the substrate into the cell is rate limiting, and the monokinetic becomes a zeroth-order reaction. It is independent of the substrate concentration. In a steady state CSTR, the microorganisms slow down their growth by depleting their environment of substrate until the growth rate μ is the dilution rate d or the inverse of the residence time. This is only possible when the microorganisms can grow much faster in free growth than they have to grow in the CSTR. When this is the case, the digester operates in the chemostat region, where a small change of residence time will only lead to a small change of the concentration of undigested substrate in the digester. When the residence time is reduced, the microorganisms will have to speed up their growth and eventually reach the turbidostat region, where a small change of residence time will lead to a significant change of substrate and biomass concentration. This region is called turbidostat because it's only possible to operate under these conditions by applying a feedback control loop, for example a turbidity measurement, and adjusting the residence time to keep the biomass concentration stable. It does not make sense to operate an AD plant in this region. After all, we are aiming for a complete conversion of feedstock. The concept is still important for the biogas process, as it will lead to acidification. If the residence time is lowered too much, the slower growing acetogens and methanogens will cross their washout point and will be flushed from the reactor. In the second presentation, we investigated the hydrolysis reactions. These reactions often show a first-order characteristic, meaning that the reaction rate depends on the substrate concentration to the power of 1. The higher the substrate concentration, the faster the reaction. When we integrate this approach for a batch reactor, we get the characteristic first-order exponential decay of the gas production, visible in biomethane potential or BMP tests. We can use the BMP tests to quantify the reaction constants. Feedstock rarely consists of only one type of molecules. Often, we see fast and slow first-order reactions running in parallel in a BMP batch test. These tests can be fitted by the superposition of 2 gas production curves with different reaction constants and different biogas potentials. When fitting the reaction constants and potentials, ignore the first couple of data points in the time series, as they contain information about the lag and acceleration phase, which is merely of academic interest, if at all, as the lag phase is a property of this individual batch fermentation and not of the feedstock or the culture used in the experiment. The lag phase does not matter in a continuous fermentation. Do not use empiric but mechanistic models to model the gas production and then combine the mechanistic model with a reactor model, for example a steady-state CSTR or a reactor cascade that describes the biogas plant to predict the performance of this AD plant. Do not confuse the reaction time in a batch with the hydraulic residence time in a continuous fermentation. These are not the same. The reaction time in a batch is the time since the start of the experiment, and since then, no new feedstock has been added and no undigested feedstock has left the reactor. The hydraulic residence time is the average time a fluid element spends in the reactor. Some elements leave before and some after the residence time. To predict the performance of a continuous reactor from a batch BMP test, use reaction modeling for a batch to identify the parameters, and then use the reactor model of the CSTR to predict its performance. The curves look different. The batch reactor is described with an exponential function, whereas the steady-state CSTR is described by a hyperbola. If a batch reactor reaches 90% conversion after 20 days, a single-stage CSTR must have a residence time of 78 days to have the same conversion efficiency. This mistake is very common in the AD industry and has caused millions in damages. A better alternative to fitting kinetic constants to BMP tests is to monitor the decay of the gas production of a CSTR after a feed stop, for example when the feeding system breaks down in a commercial biogas reactor. The observed reaction rates in large-scale reactors are higher than those observed in BMP batch tests. The reason for this is that the perceived first-order hydrolysis kinetic is actually a second-order kinetic. The reaction is substrate plus water reacts to biogas and biomass needs to be present for the reaction to occur. The biomass concentration in a running continuous biogas reactor is usually much higher than in a batch reactor when external inoculum must be provided. Additionally, small-scale lab reactors are often diluted as they struggle with higher viscosity caused by high substrate concentrations at the beginning of the batch experiment and large particle sizes compared to the reactor size. The substrate consumption rate is minus Ks for a first-order reaction, minus K prime xs for a second-order reaction, and minus mu max x over yxs times s over s plus Ks for a monokinetic. The biomass concentration x is roughly constant throughout a batch experiment and can be pulled into the reaction constant. This creates a pseudo-first-order reaction where the first-order reaction constant is a constant at least in the short term and within one batch experiment, but it will vary if the biomass concentration is changed. Like the monokinetic, but unlike the first-order reaction, the second-order reaction shows a washout point in a steady-state CSTR, meaning that the second-order reaction shows no conversion for very small residence times when the microorganisms cannot keep up and are flushed from the reactor. However, since AD plants are designed to operate in the chemostat region, the first-order reaction approach is often good enough to describe a biogas reactor and has been widely adopted, for example in the anaerobic digestion model 1 or ADM1. I have a separate presentation on the ADM1. Select an appropriate kinetic model for the type of feedstock and the application that requires modeling. First order for fibrous feedstock and high residence time digesters. Monokinetic for readily available feedstocks that do not require hydrolysis. Second order reaction for fibrous feedstocks in reactors with short residence times. Acetic acid is the substrate for acetoclastic methanogens, and the conversion of acetic acid to biogas follows a monokinetic. Acetic acid is not inhibitory to acetoclastic methanogens. Only the uncharged form as acetic acid is bioactive. Acetate is inert. Propionic acid does inhibit acetoclastic methanogens. Again, only the uncharged form is toxic, propionate is inert. The accumulation of acids in the reactor will lower the pH, which shifts the dissociation equilibrium towards the bioactive species. At lower pH, acetic acid becomes more available and propionic acid becomes more toxic. Free ammonia is both a substrate and an inhibitor for methanogens. However, methanogens require only very little ammonia to growth. Only the uncharged form of total ammonia is bioactive. Ammonium, or NH4+, is inert. The dissociation equilibrium of ammonium is temperature dependent. At 25 degrees Celsius, the pKa value of ammonium is 9.25, but it drops when the temperature rises. At 40 degrees Celsius, it is 8.8, and at 55 degrees Celsius, it is 8.4. At thermophilic conditions, ammonia is much more toxic than at mesophilic conditions. Higher total ammonia nitrogen will increase the pH of the digestate, shifting the dissociation equilibrium from the inert ammonium to the toxic ammonia. This reinforcement mechanism makes ammonia a very potent inhibitor for the methanogenesis. Methanogens will try to compensate higher total ammonia nitrogen levels by increasing the acetic acid concentration in the digester. However, at some point the accumulation of acetic acid has a negative impact on the acetogenic bacteria in the digester. Acetogens are grouped into homo-acetogens that make only acetic acid and syntrophic acetogens that make acetic acid and hydrogen. The latter oxidize higher volatile fatty acids like propionic, butyric, and valeric acid to acetic acid and hydrogen. These reactions do not generate a lot of energy, and the reaction becomes thermodynamically inhibited when the reaction products, most notably hydrogen, and to a lesser extent acetic acid, accumulate in the environment. As a consequence, syntrophic acetogens can only grow in symbiosis with methanogens. The oxidation of propionic acid is most sensitive to high hydrogen partial pressures. Oxidizing propionic acid produces 3 moles of H2, whereas the oxidation of higher VFA only produces 2 moles per reaction. At low hydrogen partial pressures, the hydrogenotrophic methanogens will become thermodynamically limited. The hydrogen partial pressure in a biogas reactor is tightly controlled to the so-called thermodynamic hydrogen window, which is roughly between 10 to the power of -4 and -6 bar. To assess if a reaction is viable, we calculate the Gibbs free energy of reaction from the standard free energy of reaction, the stoichiometric coefficients, and the activities of the adducts and products. The free energy of a reaction must be negative for it to occur. At high hydrogen partial pressures, the acetogenic reactions become endergonic, and the acetogens can no longer grow in the reactor, which will lead to the accumulation of higher VFA in the digester. In the biogas reactor, all reaction steps run concurrently and influence each other, but often only one of the steps is rate limiting and should be focused on. In the acetogenesis, fermentative bacteria produce hydrolytic enzymes and break down complex biopolymers to CO2, H2, and higher VFA. This is the rate-limiting step when larger fibers are degraded. Use first or second order kinetics with various substrate fractions to describe this reaction. Syntrophic acetogens convert higher volatile fatty acids to acetic acid and hydrogen. This step is rate-limiting when acids or hydrogen accumulate in the digester. Methanogens convert acetic acid and hydrogen plus CO2 to methane. Methanogenesis is modeled as a monod kinetic and is rate-limiting when high concentrations of VFA and ammonia are present. Other factors like temperature, pH, salinity, trace elements, and macronutrients can cause additional inhibition or limitation by reducing the maximum growth rate μmax. A slight inhibition or limitation will push the washout point to higher residence times, but as long as the washout point is far enough from the current residence time, the reduction in μmax has almost no impact on substrate conversion. Only when the growth restriction becomes so severe that the plant operates near the washout point, it will become unstable and volatile, fatty acids and hydrogen start to accumulate. If this happens, increase the residence time by reducing the feeding. This concludes my mini-series on microbial growth in the AD process. The biotechnological aspect of the AD process is often overlooked in biogas textbooks, and I wanted to highlight the implications of bioreactor fundamentals on the biogas process. For those who are interested in more details, I have a presentation on the ADM1 model which goes into a bit more detail on the metabolic pathways. Until next time on Biogas Basics.